Hello friends, I am SSV Vishnu Botla from Chandu Biology classes. In last session, we discussed briefly about what is TSSET examination and clear cut about TSSET notification also. In this session, we are going to discuss the syllabus of TSSET examination and also how to prepare for the TSSET examination. Yeah, these are some of the students who got coaching from our Chandu Biology classes and they qualified in the TSSET 2022. Now coming to the examination, first of all, we don't do hard work. We have to do the smart work and we work intelligent. We are well familiar that the TSSET examination of life sciences, which mainly consists of 13 units. In that 13 units, compulsory we have to prepare for 10 to 11 units. From wherever background you have to compulsory prepare this 10 to 11 units to crack this TSSET examination. Yeah, first coming to the unit 1, that is the molecules and their interaction relevant to biology. Otherwise, we can simply call it as a biochemistry. These are the important topics we study in biochemistry. First of all, in biochemistry, we study the chemistry of biomolecules and also their metabolism. What are the biomolecules? Those are carbohydrates, proteins, lipids, and nucleic acids, vitamins. These all are comes under this biochemistry and we have to study this very briefly and elaboratively. And these are the important concepts we have to read compulsory in the chapter 1. That is the stabilizing interactions. In that, hydrogen bonding, electrostatic interactions, van der Waals forces, and hydrophobic interactions. Coming to the next subunit, that is amino acids, their classification, structure, and next isoelectric point and pK values. In that, we have to compulsory read this Ramachandran plot. After that, protein folding, thermodynamics, denaturation, and biological functions. The very most important topic is enzyme kinetics. In this, numerical questions are asked graphical. And next, inhibition process and Michaelis-Menten equation. After that, cellular respiration. We all well familiar from our BSc. That is, we studied this metabolisms. That is glycolysis, Krebs cycle, etc., oxidative phosphorylation. So in this, what we have to study, not only just the pathway, and also we can study the regulations and what are the inhibitors of these various cycles. Next one comes nucleic acid, that we have to study their chemistry and what are the types of nucleic acids and structure of purines and pyrimids. Next, coming to the fatty acids. In that, we study different types of lipids, bonds which are involved, and beta oxidation of fatty acids, everything. After that, some numerical problems related to the thermodynamics. These are the main important concepts which are asked under this biochemistry chapter. Coming to the second unit, that is cellular organization. In this, compulsory we have to learn each and every organelle. Firstly, we have to read the major organelles, that is nucleus, mitochondria. After that, chloroplast. And next, some of the organelles like peroxisomes, lysosomes, enzymatic components of the above organelles, and diseases. Mainly, what we study, just detailed description about the organelle. But what they asked in every time in examination, along with the above organelle, they asked what are the enzymes present in it. And also, what are the functions they performed. And also, the deficiency of these enzymes causes various types of diseases. Those diseases they are going to ask in the examination. So we have to prepare in that way. And next, structure of heterochromatin, euchromatin, and transposons. After that, another subtopic, that is cell cycle regulation. Before the cell cycle regulation, we have to study the cell cycle completely, and after that, how it is regulated. In that, CDKs and CDCs, which are involved in GS1, S, and G2 M phases. After that, numerical and graphical questions based on the microbial physiology. Coming to the third chapter, fundamental process. First of all, what are the fundamental processes? We are very well know, that is a DNA replication, after the transcription, translation, and also one more thing comes under fundamental process, that is the DNA repair mechanisms. These are the main we have to read under this fundamental processes. We know that replication, transcription, and translation, everything we can we read in either prokaryotes and also eukaryotes. Every time in what they asked, every time they mainly focused on the eukaryotes, and also we have to study this prokaryotes also. It is also same important as the eukaryotes. Before the replication, what we have to study? What are the enzymes? And what are the tools which are required in the DNA replication? Here tools means nothing but the enzymes which are involved in the initiation, elongation, and termination of DNA replication. After that, transcription, translation. Next, what are the antibiotics that block these processes? And next, name of these enzymes and their functions and mechanism of action which are involved in the MMR, NER, and BER, DBR. What are all these? These are the repair mechanisms. Next, RNA processing, that is transcription and different types of RNA structure, function, and the site of synthesis. After that, the translation factors. In the translation, in what is the initiation, elongation, and termination steps, both in eukaryotes and prokaryotes. The simple way we study this entire chapter in a tabular form. That is, coming to the replication, we study in both prokaryotes and eukaryotes. What are the enzymes required here, and what are the enzymes required here for the replication? After that, on the mechanisms, how it is get initiated and how it is get initiated, elongation, and next termination. Main thing what we have to do is comparison between the prokaryotes and eukaryotes in the first step, that is a replication. After that, say like same wise we have to do it for transcription, and next we have to do that for the translation also. Yeah, coming to the fourth chapter, that is cell communication and cell signaling. Among the syllabus, the two biggest chapters, that are 4th chapter and 13th chapter, and these two chapters have so much weightage compared to other chapters. Why these two are biggest chapters? Because of the based on the syllabus. There is a huge syllabus. 4th chapter, it mainly consists of a different subunit. That is, first of all, cell signaling, and next cell communication, and next host pathogen interactions. After that, immunology, cancer biology. Every subunit is very important. Yeah, coming to the host pathogen interactions. First of all, what we have to study in that subunit? Name of the pathogen causing disease. Next, mode of transmission, and next, disease caused by it, and next, pathophysiology. After that, the subunit cell signaling. In cell signaling, what we have to study? Various cell signaling pathways. That is GPCR, MAP kinase, JAK-STAT, insulin pathway, hedgehog pathway, Wnt signaling, Notch, delta, beta-catenin pathways. After that, in this chapter only, we can study the secondary messengers. various secondary messengers. We all know calcium, IP3, nitric oxide, etc. Coming to the cell communication. In this cell communication, first of all, various types of CAM molecules and also their detailed structure we have to study. And next, functions and calcium dependent or independent molecules. Next, cell matrix interactions. Structure and function of collagen, keratin, fibrogen, nidogen, chondrin, sulfate. Coming to the cancer biology, this is the most important topic. And in this cancer biology, what we are going to study is the definitions of a benign tumor and malignant tumor and the proto-oncogenes. And what are the mechanisms of angiogenesis and metastasis, and special emphasis on the genes like P53, P21, PRB, etc. And also what we have to study, anti-cancer drugs. Coming to the chapter that is immunology. Immunology, we study what are the immunities, that is innate and adaptive immunity. And after that, all WBC cells and their function. Next, receptors of B cell and T cell and MHC molecules, structure and function of 5 types of antibiotics and preparation of monoclonal antibodies. Yeah, coming to the unit developmental biology. What is developmental biology? Simply from the gametogenesis, fertilization, cleavage, gastrulation, and organogenesis. After that, organism is formed. These all steps comes under the developmental biology. For each and every step, if we take this fertilization, we study the fertilization process in various animals. And coming to this cleavage, we study this cleavage in different animals as a models. So comparative study is very, very important in this developmental biology. We compare each and every step, that is cleavage in sea urchins as well as in humans. Likewise, coming to the gastrulation, we study this gastrulation in frog, sea urchin, human. Like that, each and every step we have to study in this developmental biology. From whichever background you are from, compulsory we have to study the chapters, that is from 1 to 6 chapters. That is biochemistry, second one is cell biology, third one is molecular biology, and next fourth one is fundamental process, that is cell signaling, communication, cancer, immunology, and fifth one is developmental biology, and sixth chapter, most important chapter, that is plant physiology. If you are from zoology background also, we have to study this plant physiology because it contains more weightage. Yeah, these are the concepts we have to study. That is, we already discussed in fertilization, cleavage, and next gastrulation, next organogenesis, and how the organism is formed. Coming to the 6th unit, that is plant physiology. In plant physiology, there are so many subtopics. First one is photosynthesis, and next nitrogen metabolism. After that, secondary metabolites, and next sensory photobiology, stress physiology. After that, transport in plants. Every topic is most important. Under photosynthesis, we study what are the pigments involved, and next, what are the inhibitors of ETC, CO2 composition, and pathways, that is C3 and C4 pathways, and what are the differences between the cyclic and non-cyclic photophosphorylation. Coming to the mineral nutrition also included under this. For this mineral nutrition, we have to based on the 12th NCRT. Okay, in that mineral nutrition chapter is there. Under this mineral nutrition chapter, we study the what are the micronutrients and what are the macronutrients. They asked this micro and macronutrients and various functions. After that, transport in plants, that is solute transport, what are the theories, cohesion tension theory and bulk flow mechanism, and detailed structure of xylem and phloem. Coming to the plant hormones, we all well familiar about plant hormones, that is auxin, gibberellic acid, abscisic acid, and next cytokinins. After that, ethylene. In this, we have to study their functions and signaling pathway, and which are antagonistic to each other. Like that, we have to study each and everything in a comparative way to remember very easily. After that, nitrogen metabolism. In nitrogen metabolism, it consists of various enzymes which are involved in different steps of nitrogen metabolism, just like nitrogenase enzyme. And next, we have to mainly focus on enzyme involved. First of all, what are the steps involved in the nitrogen metabolism? Steps involved in nitrogen metabolism. After that, what are the enzymes involved? Next, what are the bacteria involved in nitrogen metabolism? Free living and symbiotic bacteria involved in nitrogen fixation. Different types of genes also we have to study. This is a very important one. Whatever chapter you take, compulsory we have to study what are the genes, drugs, antibiotics, inhibitors, signaling pathways. Compulsorily we have to focus on this. Coming to the structure and also function of enzymes, that is nitrogenase, nitrate reductase, and nitrate reductase enzymes. Yeah, coming to the secondary metabolites. In the secondary metabolites also, we have to study the types. After the types, we have to study the biosynthetic pathway and also functions. Coming to the sensory photobiology. Sensory photobiology, it mainly consists of 3 types. That is a phytochrome, cryptochrome, and phototropin. Among these 3, we have to make a chart in a comparative way. In that, we have to study structure and functions. Mainly, we have to focus on the functions and some of the concepts. That is a photoperiodism, circadian rhythms, and nyctinacity. Like that, we have to study some concepts along with this comparative table also. Special emphasis of biological clocks. And next, stress physiology. Coming to the unit 7, that is animal physiology. In animal physiology, first of all, we have to give the main preference to the subunit, that is endocrine system. After that, respiratory system, excretory system, nervous system, blood, and cardiovascular system. In endocrine system, they've mainly given the hormones and their functions. And next, in respiratory system, they can ask in a statements or either graphical model. Coming to the excretory system also, some little bit of a numerical part, and like that statements also they given. Yeah, in endocrine system, first of all, we have to study that hormone, from which gland it is going to released. And next, what are the amino acid precursors and deficiency disorders? This is the way we have to study each and every system. And next, respiratory system. In this respiratory system, how mechanism of gaseous exhales, and next, respiratory volumes. We all know these numerical values of tidal volume, IRV, ERV, etc. And next, mainly we have to focus on hemoglobin. Coming to the blood vascular system, we have to compulsory study this ECG. Every time they ask questions from the ECG concept. Coming to the nervous system, Resting membrane potential and action potential, they are asking frequently. And next, excretory system. In this excretory system, they mainly focus on the solute ions and essential elements that reabsorbed in PCT and DCT and loop of Henle. And next, symporter, antiporter present on the these parts. After that, sense organs are next important to all these subtopics. In sense organs, what we are going to study, that is the inner parts of the ear. And next, eye. And last, digestive system. In this digestive system, every time we study just like just organelles, that is a mouth, like a pancreas, a small intestine. But these are not important. What are the important concepts in this organs is, we have to study in stomach, there are different parts. In that different parts, what are the enzymes or juices and released. And next, what are the pH values. And next, it contains various juices, intestinal juices. In that, what is the composition, and if there is any deficiency of vitamins, what are the diseases are occurring, and what are the disorders facing, like that we have to focus. Yeah, coming to the inheritance biology. If we don't want to, anyone don't want to study this 7th chapter, we can easily alter it with the 12th unit, because we have two options, that is either we read 7th chapter or 12th chapter. And next also, either we focus on 9th chapter or 11th. We have to select any two from these 4 chapters, and remaining all are compulsory. That is from 1 to 6 compulsory, and next 8, after that 10 and 11. First of all, we have to prepare very well in this 6 chapters. After that, genetics, and next ecology, evolution. At last, we have to focus on the 2 chapters from this from among these 4. Coming to the genetics, that is inheritance biology. In this, various principles are there. We all know that is a Mendelian principle. Next, in that various concepts, Mandela's are there. Dominance, segregation, independent assortment. After this, some concepts are there, that is codominance, incomplete dominance, and gene interactions, pleiotropy, etc. After that, genetic transfers. What are the methods involved in this genetic transfers? That is a conjugation, transduction, and sexduction. And next, mainly we have to focus on this pedigree analysis. Every time they asked one question from this pedigree. And next, polygenic inheritance and QTL mapping. Next, types of homologous and non-homologous recombinations. Every student mainly focus on this up to these topics. But what we have to know is, every time they ask questions from this concepts. Why? Because to increase the toughness, and also to increase the difficulty, to increase the difficulty of asking question. So they have to mainly focus on this pedigree analysis and homologous and non-homologous recombination, linkage mapping. From this, they ask questions more frequently. And next, chromosomal alterations. Very, very important topic is chromosomal alterations in the deletion, duplication, inversion. In this, they asked mainly focus on the statements, and also we have to focus on pictures or diagrams, and also examples. Coming to the 9th unit, we can opt either 9th unit or 12th unit. if you are from zoology background, we can easily choose this 9th unit. Or you are from any other background, you can easily choose this 12th unit. From these two, we can either choose any one of these. In this, we mainly focus on the phylogenetic tree cladograms. And next, mostly it is based on the NCERT level. They ask questions mainly from the NCERT level. For that, we have to mainly focus on invertebrate phyla and also vertebrate phyla. After that, types and mode of nutrition, types of reproduction, that is a vegetative, asexual, sexual mode of this 5 kingdom classification, that is a Monera, fungi, plantae and animalia, protista. After that, the parasites and the pathogens of humans. And next, rare endangered species and their conservation strategies. Now they are mostly asking the questions from this. What are the endangered species and what are the conservation strategies? What are the rare species? Like that, they are frequently asking questions from this topic. Coming to the ecology. Ecology is a very most important concept. In this ecology, what we have to study first is the subtopic, that is habitat and niche. After that, we have to study related to the population. Group of population, we called it as a community. After that, ecosystem. In that way, we have to study. First of all, habitat and niche. After that, the types of succession. Next, ecological rules. Here, various rules are there. That is Allen's rule, Glogger's rule, Bergman's rule. In this, they ask the statement type of questions and some typical questions. After that, population ecology. In population ecology, some numerical questions are there, and here we find some formulas. So we have to focus on this along with the statements and also theory. What is a population density? And next, here diagrams also very, very important. What is a uniform, grouped and random? Next, mark recapture method. Last time they asked this mark recapture method in the CSIR examination also. Types of dispersion. And next, population growth curve, metapopulation, 3 types of survivorship. Each and every concept in the ecology is very, very important. They touch each and every corner from this chapter. Here R values and P values are also most important. Coming to the community interactions, that is symbiosis, commensalism, amensalism, parasitism, competition and predation. Here examples also we have to remember. After that, Loca-Volterra model, very, very important one. And after that, types of pyramids, ecological pyramids, energy, biomass, number in terrestrial, aquatic and also grassland ecosystem. Yeah, this is also very, very important. That is a lentic and lotic ecosystem. In this, various concepts are also there. That is a eutrophication, biomagnification, bioremediation, phytoremediation, and types of diversities. And next, pollution. In this pollution, we have to study what are the causes of air pollution and what are the gases causing the air pollution. And next, for water pollution, we have to mainly focus on the BOD and also COD. These are most important. Frequently in a CSIR also, they are asking these questions. The list of Project Tiger Zones, national parks, sanctuaries, and biosphere reserves. First of all, definitions. And next, examples. Conservation strategies. In this endangered, critically endangered, we have to remember the examples for examples of animals under this endangered category, vulnerable category, near threatened, like that. Yeah, coming to the evolution. In evolution, first of all, we have to know the theories of evolution. In that, Lamarckism and Darwinism, Lamarckism, Neo-Darwinism, like that, there are various theories. After that, experiments done by the Operon and Halden, experiment of Miller. And next, geological time scale, which is a very most important. Here, geological time scale is very, very important. We have to remember this. After that, phylogenetic trees are more important. And next, genetic drift. Definitions of convergent, divergent evolution. Here, we have to remember the statements. That is a concept and also the examples. And what are the examples of homologous and analogous organs? And next, Hardy-Weinberg equilibrium. Numericals comes from this Hardy-Weinberg equilibrium. And next, altruism, kin selection and reciprocal altruism. This is also important. These concepts are more important. In this, they can ask every type of question, that is statement, concept-based or experimental, or either they can ask a numerical. For this numerical, they focus on a Hardy-Weinberg equilibrium. Coming to the applied biology. For this applied biology, we can choose either 7th unit or 12th unit. We can focus on either 7th unit or 12th unit. And coming to the methods in biology. Yeah, this is one of the huge chapter in our syllabus. Under this 13th unit, we study various type of techniques. That is chromatography, centrifugation, like that agarose gel electrophoresis, everything, every technique we study under this methods in biology. And also we study this biostatistics. Yeah, biostatistical methods. In this, so many formulas are there we have to remember, and also numerical type of questions. The topics are regression and correlation, t-test, ANOVA, chi-square test, and probability distribution. After that, we focus on electrophysical methods. So that is ECG, EEG, MRI, fMRI. And next, microscopic techniques. In microscopic techniques, we study the freeze etch and freeze fracture methods. And next, histochemical and immunotechniques. We study under this ELISA, RIA, and Western blot, flow cytometry, FISH, and GISH. Next, these are very, very important. That is RFLP, RAPD, and AFLP. All techniques are very much important. They interconnect this chapter to other topics. In biophysical methods, they ask related to the UV, CD, NMR, ESR, and X-ray diffraction. Coming to the vectors, different types of vectors used in rDNA technology. First, we have to give the preference for rDNA technology. In that rDNA technology, we study various types of vectors used in the rDNA technology, like that. Plasmids, cosmids, and BAC, AAC, etc. And isolation and purification of RNA and DNA. After that, gene knockout and knock-in experiments also. Yeah, this is the complete syllabus and what are the important topics we have to study in this. So, by knowing all this, we have to plan how to prepare for the exam in a smart way. If you want to join, you can easily join in our Chandu biology classes by simply contact to the number, or you can email us.